The universal energy currency for our cells is adenosine triphosphate, also known as ATP. In eukaryotes, a major portion of ATP is generated in the mitochondria. In a brief summary, cytochrome C oxidase is an important enzyme in mitochondria related to the process of oxidative phosphorylation. Oxidative phosphorylation is the process in which energy transformed in the breakdown of various carbohydrates, fats, and proteins is used to synthesize ATP for further use. It is involved in cellular respiration, which is of the utmost importance because other methods of ATP generation only yield around 2 ATP, while oxidative phosphorylation yields around 32 ATP. That's 16 times more than other methods. Cytochrome C oxidase are composed of 3 subunits made from mitochondrial DNA, and between 8 to 11 subunits made from nuclear genes. 2 of the subunits, COX-1P and COX-2P, require the cofactors heme groups, an iron-containing prosthetic group in COX-1P, and copper ions for both COX-1P and COX-2P. These 2 subunits are responsible for the transport of protons from the matrix to the mitochondrial intermembrane space. Cytochrome C oxidase is described as a proton pump because the enzyme catalyzes electron transfer from cytochrome C into molecular oxygen, reducing the latter into water. In simpler terms, the cytochrome C, a water-soluble hemoprotein, is the substrate for the enzyme. The cytochrome C transports electrons into the active site of the enzyme. The enzyme then requires the cofactors, a heme ion and a copper ion, to reduce O2 into water molecules, therefore allowing the mitochondria to complete cellular respiration, producing ATP for use around the body. Now, what are some inhibitors for the cytochrome C oxidase? Cyanide, azide, and carbon monoxide are inhibitors for the enzyme due to them binding to either the active site or the enzyme itself, causing it to not work. Another inhibitor for cytochrome oxidase is having no oxygen to work with. For example, high-intensity exercise or having a heart attack will cause the enzyme cytochrome oxidase to shut down. This in turn causes ATP yield to sharply decrease. When cytochrome C oxidase is faulty or mutates harmfully, it will cause a defect in the process of oxidative phosphorylation. This is responsible for a multitude of genetic disorders that impact on tissues and organs with high metabolic demands or dependency on mitochondrial metabolic pathways. Specifically for cytochrome C oxidase, there are a number of mutations in assembly factor and structural genes that causes forms of Leigh syndrome, a severe genetic neurological disorder. Leigh syndrome develops in 3 stages. The first stage is between 8 and 12 months and involves the symptoms vomiting and failure to thrive, which is when weight or rate of weight gain is significantly below that of other children of similar age and sex. The second stage is in infancy. The symptoms include the loss of motor ability, eye problems, and respiratory irregularity. This stage occurs between 2 and 10 years of age. The symptoms at the third stage include hypotonia, meaning decreased muscle tone, and feeding difficulties. Another deficiency called the mitochondrial complex 4 deficiency, also known as cytochrome C oxidase deficiency, is caused by defects in the enzyme. The symptoms of this deficiency vary from person to person. The people who are mildly affected show symptoms like muscle weakness and hypotonia. The more severely affected individuals show problems in multiple body systems. Symptoms include severe brain dysfunction, a type of heart disease that enlarges and weakens the heart muscle, an enlarged liver which may cause liver failure, and a buildup of lactic acid in the body, which can cause nausea and an irregular heart rate. Many people who have this deficiency do not survive past childhood, while those with milder symptoms can live into adulthood. Many people who have this deficiency can also have a more severe kind, known as Leigh syndrome, as discussed before. Mutations in this enzyme also cause other rare syndromes that result in neurological disorders, along with impacts on the heart, liver, kidney, and digestive systems.